For our sec for our second second talks for the molecular connectivity online series, or MCOS. My name is Cindy from New Jersey Institute of Technology. So I'm part of the group, the working group of the molecular connectivity. In case you are new here, so this working group is consists of a diverse background of researchers from medicines, neuroscience, and physics. The aim is to advance molecular connectivity as a tool to study brain connectivity. We organize this online series as a platform to exchange ideas and promote research on molecular connectivity. So we plan to host this online series monthly for every third Fridays of a month. You can go to our website, subscribe to our newsletters, and follow our Twitter or LinkedIn. Today, I'm pleased to introduce Dr. Christian Habeck from Columbia University in the city of New York. Dr. Habeck was trained as a physicist in Germany and the UK in 1890s, and then switched to neuroscience as a postdoc. And after some computational modeling at the Neuroscience Institute of San Diego, he turns to study human neuroimaging analysis and comes to Columbia University ever since. In today's talk, Dr. Habeck will introduce the multivariate neuroimaging analysis for neuroimaging data for nerves and novices. I would also like to introduce Dr. Deborah Peretti as the moderator for today's discussions. Dr. Peretti was also trained as a theoretical physicist in Brazil and switched to the field of PET imaging data analysis in the Netherlands for her PhD. And then she becomes working for PET quantification and molecular connectivity ever since. And now she is a postdoc fellow for the University of Geneva. Once again, thanks everyone to coming, and please welcome Dr. Habeck for his talk. Thank you.
Okay, thank you everybody for coming, and I know there'll be a wide range of expertise in the audience, and my hope is that everybody will get something out of this. There's a little bit of very basic introduction, and then I'm going to use a technique that's called the scaled subprofile model, and the inventor of this technique is actually, I believe, in the audience. So if you have any questions, particularly some about the history of what correlational modeling of PET data was in the 1980s, we could talk about this later. I'm not going to go about the historical background very much. So I always enjoy talking about this, and I think particular principal component analysis, but any multivariate technique, there's always something to discover, and it's a very deep topic that sometimes is still full of surprises. So oh yeah, then I also want to give thanks to the Molecular Connectivity Working Group, and I've highlighted his name the most because he kind of burst into my life in 2018. He's a force of nature, and I'm glad he did, but all the other people here, very talented individuals, very, very happy to be working with them, and looking forward to the future. So then I also want to mention, I always say this, I once heard this at a talk, which I like very much. There is no I in fMRI. They call it the Buddhist mantra of cognitive neuroscience. And for PET, there's literally no I in it, so it works even better. So there is no I in PET, and all these people, ones in sort of more bolded font, current colleagues, and all the other ones I have sort of interacted with and am interacting with profitably, and they're not based directly in our group, but all of these people have contributed. So I'm going to give a very opinionated introduction that's very fast, and I find it interesting because not too long ago, multivariate analysis was kind of somewhat frowned upon. I remember this when I first came as a postdoc to Columbia, and there was a strange kind of culture war going on where multivariate analyses were kind of pitted against univariate analysis, and at that time SPM was kind of the very dominant mode of fMRI analytics. And there were these sort of allegations, or almost sort of sort of accepted beliefs that univariate proponents would always bring up that multivariate analysis had no false positive controls, that often the non-parametric inference you needed because there were many more observations, many more observables than observations, that they were highly suspect, that they were hard to interpret. And I remember at a talk that wasn't so long ago, I compared multivariate and univariate analyses on a biomedical engineering professor at Yale. He told me afterwards, yeah, I really enjoyed your talk, but I don't really understand what, it's so strange to have this fight or this dichotomy of univariate versus multivariate. I mean, it's sort of like you're comparing apples to oranges. They're both just different aspects of the signal. It's so strange to perceive a conflict between them. And I had to agree with him. I think he was absolutely correct. At the same time, I was kind of making a career out of this misunderstanding, so I had to acknowledge too. Also, it's interesting that at that time already, multivariate analysis had been performed already for decades. And you could see some of these names: Barry Horowitz, Stan Rappaport, Jim Merlos, Stephen Strother, Randy McIntosh, David Eidelberg. Many of you know his work, I know. So people have been doing multivariate analysis in resting FDG PET already for a long time with very good sort of results. So it shows you how the community sometimes can be fractured, and the fMRI community was sort of on a totally different wavelength, you know. And that's, I think, also the what you can discover today, where sometimes whole fields are talking past each other, and you know, sometimes rediscovering things that already have been well established, possibly decades earlier. And I would say today, I mean, multivariate analysis, of course, is firmly in the mainstream. You know, in the mid 2000s, I think with the GIF toolbox, you know, and FSL, ICA became very prominent also because of good software support. And today, I think none of this skepticism is there anymore for multivariate analysis. It's actually the opposite. Possibly, it's gone a little over the rails with highly derivative frameworks that sometimes become so complicated and sort of like clear why it's more intellectual ownership than scientific illumination. So I want to say the spirit of this talk is that we molecular connectivity techniques strive for an informed use of connectivity approaches. I'll be focusing mainly on PCA today. And we want to follow this great tradition, and we want to make it as complicated as it needs to be, but no more. So we want to avoid these anti-Occam's razor tendencies that can sometimes be observed. So just, and I always show this just with some very simple theoretical motivation. If you have a data array, and this is an actual fMRI data array, although the data set was updated since, but imagine you have a data array, and these are voxels as rows and participants as columns, right? And usually all the situations I will talk about have many more voxels than participants. So in other words, it's a very rank deficient data array. And usually, just to recall the univariate mode of analysis, you would go row-wise and you would run a design matrix, you know, that encodes, you know, task effects or even subject performance, something that you're interested in, and you would run linear models for each row, so many of them, and you would usually have some kind of family-wise error control, or, you know, false discovery rate, and there's nothing wrong with doing things like that. I'm not going to cast dispersions on this mode of analysis. But it's maybe good to keep in mind, because of this rank deficiency, there are only n, if the data rank is n, there are only n possible independent things going on in the data. That's dumb dimensional analysis. And particularly if you're interested in predicting an outcome, imagine you want to predict from this high dimensional, let's call this RV, when V are the number of voxels. If you want to predict an outcome, then it's probably judicious to consider the correlation between all these voxels. That's necessarily true. After all, you can only pick n out of those, and then you get linear dependence, right? So most likely, if you come up with some prediction algorithm or classification, then this rank deficiency somehow needs to be dealt with. So that's the first argument. And this has nothing to do with connectivity. That's just dimensional sort of dumb luck. And just to explain now, any kind of multivariate decomposition, you can now see as you have a data array, you have a matrix of components, and then you have a matrix of component scores, or sometimes it's called mixing matrix. And usually what you want to do is you want to have only a few components that capture the signal as best as possible while you relegate the noise into this noise term, and you're only considering this. So this is generic. Any multivariate decomposition will work this way, right? That's good to keep in mind. The point is that you isolate all the voxel specific information into the component part and the subject specific information into the score matrix or the mixing matrix, or their different terminologies depending on what technique you're using, right? And we can do all kinds of things. I'm focusing on principal components here today, that imposes orthogonality of the columns of V and W at the same time. But of course, you could look at spatial independent components analysis, where it's the statistical independence of the components of V, so the columns here are statistically independent. You could do other things, non-negative matrix factorization, very possible. Then there are all kinds of oblique rotation schemes, right? You can rotate the components such that particular properties of the loadings are fulfilled. That's probably similar to ICA some way, but if you take Varimax or Promax. You could come up with your own, right? I mean, you can come up with your own decomposition that's motivated by some sort of regularizers, possibly also, or some other demands you make on the component loadings. And here's an example. Imagine we have 10 components we're interested in. Here would be the matrix of the actual components, but these will be real brain images. And here would be the mixing matrix or the score matrix, right? And it's transposed. So, we have 255 subjects, but n component scores. And a big part of this talk today will be the following caveat, and we're going to come back to this often. Don't confuse mathematical convenience of the decomposition with scientific meaning, right? So these techniques are wonderful. People have worked on them. PCA is more than 100 years old, and PCA and ICA, I think, have also no stochastic variability. So you apply them to a data set repeatedly, you always get the same answer, and things are maximally statistically independent or uncorrelated. But this will work on any signal, right? And so even when you submit noise, and I'm going to show some of these examples later. So keep this in mind. We're going to need outside validation in order to convince ourselves that these are meaningful, right? And some of the more uninformed use, in the early maybe 2000s of multivariate techniques could be found in the literature, where people were analyzing components only by virtue of their coming out of a decomposition, no other outside validation, and that's arguably not a very good use, or not very convincing use of multivariate decomposition. So this is often, and I don't know, maybe we can stop after I've gone through here and ask for some questions. This is maybe not super relevant. This is just one for people who are stumped by this. Don't get too hung up. This is just one thing that I think is nice to show that PCA works, because this is the sometimes called the eigen equation of PCA, and I'm not going to explain it very much, but what's nice, so these eigenvectors are in a way they have the property that you apply the covariance matrix onto these eigenvectors, they just get scaled. They don't get transformed or rotated or translated in any way, they just get scaled. That's a very nice property. And what's really nice, and I think sometimes people don't appreciate it, that you can do this in both spaces, both in the voxel space, but also in the subject space. And that's a very nice property. That means even if you have, imagine you have genetics data that are millions of SNPs, but you have 1,000 subjects, you can easily do this PCA decomposition in the subject by subject space and get the corresponding SNP loadings by projecting back. That's a very nice property. You can do everything in the dual space and then project back. That also meant you could do PCA even early when, you know, computational facility wasn't so good, because you were usually dealing with subject by subject covariance matrices, and then you could project back. Okay, and as I said already, the nice thing, so this is also sometimes called singular value decomposition. So with the right scaling, where you incorporate the eigenvalues into the score vectors, we come up here. And what's nice, as I said, for PCA is you get the same answer every time. There is no stochastic variability, just for non-negative matrix factorization, you know, even k-means, you don't have such luxury. Here you get the same answer every time, you know. So, and as I said, our objective is to somehow truncate, only consider a small subset of components and splitting off the noise. Okay, I don't know whether there are questions yet, or maybe if possible, I'd like to keep going because I have a habit to.
No questions so far. You can go ahead. I'll let you know if there's any questions.
Okay. And so then also a nice heuristic that many people probably know is the scree plot, where you plot the eigenvalues and I normalize them such that they sum to 100%. But you can see here there's a synthetic data set. I made up 10 sources of variance, so 10 sources of correlated activation. And this is totally synthetic. It's not real data. 10,000 voxels. And you can see when you do this, you get a nice kink in the scree plots, you know. So there are 10 real sources of variance in the data. And then everything falls almost to zero. And just imagine now I created random IID noise, taking that same signal, but permuting all the voxels within each subject, and so making total noise. And then you see this line here, that's roughly 1/800, 0.012, the orange line. That's also sometimes called the eigenvalue bigger than one criterion, right? So if I had normalized it to 800, my total variance in the data array, then the line would be at 1, and everything over this line sometimes is interpreted as meaningful signal, right? And so there are other ways of picking the sets you're interested in, you know, their whole fields, working on subset selection. You can also do, you know, n-fold cross validation. I often just use the Archaika criterion. That's sometimes a sort of a point of discussion and criticism also. There's perceived arbitrariness in the set of components that you pick, but I'm going to try to convince you that that isn't such a big deal. So,
before going forward, actually, we do have now a question. We have one of the members ask if you could explain again the subject space part of the PCA.
Let's try. Let's not get. Okay. So you see here, this is the eigenequation in the voxel space, right? So here we have the columns of V are really the eigenvectors in voxel space. They're real brain images. And here we have the diagonal matrix of eigenvalues. So now what I can do, I can take my data matrix and transpose it and multiply it from the left. And when you do this multiplication, you ultimately get this eigenequation. And now you have the covariance matrix not in the voxel space, but in the subject by subject space. That's this thing here. And W, these are the eigenvectors in the subject space. So if you have, say, 100 subjects, these eigenvectors will have 100 rows. That will be the actual vector. And I'm claiming, or, you know, I'm not claiming, it's, you see that this eigenvalue problem and this eigenvalue problem is very similar because we have the same eigenvalues. And these different sets of eigenvectors are related just by projection. So I hope that would suffice. If you have any more questions, you can email me later. It would be good to move on if that's okay. Right, Deborah, could we move on?
Okay. Yes, it's
okay.
fine. He said that it's fine now.
Okay. So this is a nice heuristic. And now what I sometimes like is you can actually really play with this in real data, and arguably that's maybe what I enjoy most. Simulated data are additionally good because they always work out well, so let's imagine there are 300 data points, and we have a voxel. We have a brain that only has 2 voxels, right? And just to make it really simple and allow Euclidean geometry in the plane here. So you see, this is the sample of 300 observations. And now if we do the PCA, you can see these are the eigenvectors that come out. This is the first eigenvector that's, the biggest source of variance in the data. And this eigenvector is PC2, and they're literally orthogonal. That means, in a geometric sense, there is a 90-degree angle, right? So now, what happens, and this is something that comes up often, yeah, maybe not so much now, but when I first started, this came up often. There are all kinds of techniques that do a prior tree transformation of these data. There's something called multilinear models and partially squares. People use very often still canonical correlation. I see in the literature very much. And all of these, what they usually entail is that you, prior to your multivariate decomposition, you reduce the data further. For instance, you could imagine, this is hypothetical now, but imagine I had some behavioral variables or seed variables in a particular region that I was interested in. And these 300 observations were 100 people, but in 3 different conditions, right? So imagine I somehow pre-transform, a computer correlational image with imagine these were 3 cognitive tasks, and I take the percentage accuracy as my seed and compute a correlation image. And now, rather than 300 images, I now have 3. And I scaled them just to make it really obvious. So my 300 data points now collapsed into 3. And now I'm doing PCA on 3 data points. That's the first thing maybe to keep in mind. You have radically reduced the dimensionality by this projection. And what could happen is that this basis could really change a lot. As you can see here, my original basis was, the blue principal components. They get transformed to the orange ones. You can see now this is the major source of variance, and this is orthogonal to the major source of variance. And you can see that, the brain images that come out can really be radically different, right? So now I'm not saying this is simulated data. So I'm not saying that doing these pre-transformations, radical data reduction prior to the PCA step, that they're necessarily wrong. It's just maybe good to keep in mind that you're doing something fairly radical, and you should be careful that you're not throwing out the baby with the bathwater. You are implicitly relying on the fact that the variance in this new transformed space of correlational images is in a way more meaningful or gives you better sort of signal. But that's an assumption, right? And so, as I said, I don't know if this is maybe possibly too strong, but I always have found it easier to not do these radical transformations and just leave the data untransformed and just get the PC structure and see whether you can find images that correlate with the behavioral outcome. And I'm going to show this later. Okay. Any more questions? If not, I would also press on so we can. So this
no questions.
This is a brief excursion, and it's very quick just to show how PCA works, and sometimes it's easier to see in a 2-dimensional example with pixels. But imagine you have this pattern in the data, again, totally hypothetical data. You have these two adjoining squares of strong loadings, and they're surrounded by zero loadings. And imagine we have young and old people that we, again, totally made up. So we now make up a data array where we take these subject scores, multiply it by this pattern, and then add Gaussian pixel noise. Everywhere we add, random IID noise. And then we can see if we crank up the noise value, these are arbitrary units, right? But you can see what happens if we do a univariate t-test without family-wise error correction here. You can see that once you crank up the noise, at some point you will lose any kind of difference, so there's nothing there that you can find anymore as a difference of young and old. But just to show, because of the distributed nature of the signal, PCA works reasonably well. Even under the strongest noise source, you'll get some components out there very similar to what you put in. And now the noise is incorporated into the voxel loadings, but you clearly see this adjoining square structure, right? So that's an example where PCA will work better than univariate analysis. And it's also good to see what are the assumptions. I put in a subtle, widely distributed signal. This thing might not work if there's just one small ditzel here in the picture, right? It works on a subtle but distributed signal, right? Okay, so let's move on. Oh yeah, also the nice feature of multivariate analysis also is, I mean, this is maybe obvious, but usually any pattern that you derive from a data set, you can apply prospectively into any other data set, whether you derive the data from the training sample or not, right? It's very easy to do this. You just have to be resliced in the same voxel space. So whenever we've derived a pattern, you can now test it and its association in totally new data. And okay. Now it's maybe the time. There were some recent caveats about a PCA, and oh yeah, I formulated this a little bit harsh here, but these caveats are good to keep in mind. But I think they just come from basically they address an unthinking overreach in the interpretation. They're not that novel as such. I think people understand it. So as I said before, PCA, when you apply it to a signal, you need outside validation, because even conflated signal with much noise will give you orthogonal components. And then there's another thing that I sometimes see in other contexts, often of genomics and neuropsychology, that picking single principal components, isolated principal components, is probably not a good idea. And I'm going to try to convince you of that fact. So let's imagine the following. This was a recent caveat, and I like the S.J. Scott a quote: "One man's trash is another man's treasure." So here PCA works on every kind of signal, right? Let's imagine we're in the time domain, and we have 50 people or 50 temporal signals, and there are absolutely null signals. I made them up. There's nothing in there, right? And I'll take out this record of 6 seconds, and I've smoothed this I have a moving window average of 1,000 steps. And after I do this, I get this smooth. You can see the smooth signal here. Again, it's based on these null signals. And if we look at the covariance matrix, because we only have 6,000 time points, we can look directly into the time domain covariance matrix. You see that there's really nothing here. I mean, you see the main diagonal, as you would, but there's really no interesting structure here in the covariance matrix at all. But this is what happens when we smooth it, right? Just by virtue of smoothing, we are finding some covariance. So you can find this sort of broad band here. And if you now do PCA, here I plotted the first 16. It's totally arbitrary, but they go in this kind of ordering, right? You can see that you get these they almost look like harmonics, right? They're almost like a Fourier series You can find them in increasing temporal frequency, right? And they come out purely out of noise that was smooth, right? So if you were to focus on a particular one, you know, you would really be very misled into possibly thinking that there's some interesting structure in the data, although this all was, you know, totally due to noise. And let me now look at a real world example, just another caveat about picking single principal components. You can do this, of course, for descriptive purposes, but the inferential robustness is really sort of questionable. Imagine this is I'm not going to talk about the fMRI task very much, but there are 324 people who do this task, and it's across the lifespan. They're doing a task that's a recognition task. It's a paper folding where they look at the paper that's being folded. It's punched in a particular manner, and then the unfolded paper has a particular punching pattern, and they have to pick out of an array of choices, and they get this right or wrong, and we get the percent accuracy. But imagine when we do PCA on this data set and plot the scores, how the PC scores, the subject scores, correlate with percent accuracy, we see the following, right? We see this correlation spectrum where PC5 really correlates the most. This is a logarithmic sine-weighted p-value. So PC5, the subject score, happens to correlate the most with the outcome, right? So you might be tempted to think, oh, so let me take PC5, right? Because it gives me the best correlation with the outcome measure here. But I'm going to try to convince you that this is not a good idea. First of all, let's think about inferentially what happens. How robust are these results? Let's take these data and sort of divide them randomly into 150 training data sets, 150 test data sets, and now we do PCA in each and just see how well those principal components, how well they are topographically related to one another. And you see, I mean, we get PC1 behaves pretty well, but you could see some of these other ones, they're not very robust, right? You also see the interquartile range of the correlation distribution. We have 100, I do this 100 times, so 100 times I split up the data into training and tests. And you see that these are really moving around a lot under my resampling procedure, right? It's non-overlapping samples, but these PCs really look nothing alike. They're almost orthogonal to one another. Then we do the following. We ask, how well does PC5 from the training set correlate with the behavior when I can do this in the training set directly? That's the red line. And I can also go to the test set. I take PC5 from training, project it into the test set, and see how well does that resulting score correlate with percent accuracy. And the nice thing is that the training and test set results are fairly similar, but only 46% of the time you get a significant result. Actually, that's actually a good result. Very often the fractions are far lower. So although you picked PC5, so when you found PC5 topographically really moves around a lot under resampling, you don't find a whole lot of stability. The one thing that may be good, it still works even in the test sample, but only 46% of the time, right? So that's maybe not what you thought would be the outcome if you pick PC5 on account of the full sample data, right? And I'm going to convince you, and this is where I introduced my workhorse for the last 2 decades, and Jim Moller is on the call, I think, hopefully. So we're using a technique he pioneered in 1987. And because he applied it to PET data, there's some additional complexities because PET data is multivariate, multiplicative variably, has multiplicative variability. I have additive variability. These are fMRI data. You can have negative values also. But again, we summarize this whole prescription, which in a way is quite simple. So we do PCA again on the fMRI data. And now, rather than picking single components, we're picking a set of components. And as I said, you could use leave one out cross-validation or AIC. I use those both of those. But we're making a component, we're making a pattern out of a whole set of principal components, and we're always including the first one. So imagine we pick K. We decided, oh, we take the first K principal components. Then we make a pattern using this behavioral regression. And we can also check how well this pattern works, either in the training data set or in the outside data set. We can, as I said, project it. We do this Euclidean dot product and then correlate it with cognition, right? And again, if this isn't so clear, hopefully conceptually it's clear. So rather than picking a single component, we're taking a whole slew that will protect us against some of this resampling variability. And you can see this now. So now what, oh yeah, and this is just the point. We'll get in the pattern, the subject scores. Now we apply this PCA technique, and rather than take a PC5, we're taking the first 5, PC1 to 5, right? So we're now using the behavioral variable to guide us to construct this pattern in the training set. And first of all, you could see all of a sudden now we have much better spatial reliability, right? The correlation between the training and the test set pattern is much higher, whereas before we had PC5, you saw, not much similarity, and you could see the violin plots over these 100 split samples look very broad, but taking those first 5 together, that protects us somewhat because now sampling variability isn't so important. Likewise, you could see here, oh, this shifted a bit, but now if we take the linear combination pattern and look at its prediction of behavior out of sample, that's the test part. So we derive a pattern from the first 5 principal components in the training set and then see whether this pattern, when projected into the test set, still correlates with accuracy. You can see that 93% of the time, we get a correlation better than 0.01, right? So much better sort of robustness of the brain behavioral correlation. And in the training set, we get a significance of 71%, and that's lower. It's just because we're taking into account we're using 5 principal components. Our regression assigns a parametric p-value that's lower. It takes into account 5 degrees of freedom. So anyway, so the takeaway point is don't take single principal components. Take a whole slew of them, ideally, and then construct a pattern that predicts behavioral performance. That's a much more robust way that inferentially works better. Okay, so then now I have one last part where I want to remind people just of the difference between univariate and multivariate analysis. If there are no questions, I think I can get this done in 5 minutes. I would go ahead with this.
Actually, we do have a question now. While you're using fMRI for PCA, the data set would have voxel by time points and subjects, right? So wouldn't that be a 3D matrix instead of a 2D?
No, that's a good question. No, and I glossed over this. Whoever asked the question, you're absolutely right. I have neglected to say I have activation maps. So there was FSL was being run first, and I have one observation per subject per task. So in other words, the time series has already been reduced with a linear model within subject. So that makes it easier. I only have voxels and subjects. Okay, if there's no more question, I would go on. So now I'm just taking a little more, this is in a way more standard, and it's not controversial, I think, just to to just demonstrate how the power of multivariate analysis, at least for fMRI data. And I'm going to say also later, I mean, I haven't dealt with PET data for a long time, but what I remember in my hands and other people's hands also, PET data will always better behave with better variance concentration. So the rationale for using multivariate analysis is even stronger, right? But let me go back to here. We have 290 people, and we have 3 fluid reasoning tasks. I'm not going to explain them very much. I mentioned the paper folding already. We also have matrix reasoning that's similar to Raven's advanced matrices, a classic fluid intelligence test where you, and again, it's a recognition version, so people need to pick out one pattern that best completes a matrix of 9. 1 is missing. And we also have this where people see a letter set of 5 groups of letters, and they need to figure out the rule by which these letters have been arranged. And one of the groups of the letters is violating the rule. So it's an odd man out. So we're assuming that the cognitive process underlying this is the same. So we stack them or concatenate them. And the cognitive outcome, again, is the fraction of the correct responses. So we do our usual scale subprofile model technique, which works very well. I've explained it already. But we compare it also, we want to compare it to univariate regression. And here we just do voxel-wise regression, where we have the outcome measure, where the percent correct, and we fit voxel-wise these linear models that will give us brain maps of the slope and the intercept. And for the out-of-sample prediction, we just have one prediction for each voxel. And what we do is we just form a vote. So we'll take the logarithmic p-value or an f value. We average all these predictions according to how significant they were in the training sample. And then in any test sample, we can apply the same model, the averaging, and we can come up with a prediction of cognition in the held out data, right? And our sampling frame, again, is somewhat arbitrary, but we take 600 test data set, training data set. We have 100 test data sets. And so we do comparing the SSM technique, that's our PCA thing, with this univariate vote predictor. And you can see that you can see these violin plots or interquartile lines. You can see that we're doing much better on average, or on the median. We predict in these held out data, we predict accuracy, about with 20% of the variance, right? So you could now ask, well, is this maybe, how about if you have avoid the rank deficiency, make it less bad, and just pick 500 randomly sampled voxels, right? Instead of 25K, let me pick 500 only. Does PCA still work better? And you can see, although PCA is suffering somewhat, the univariate vote predictor doesn't suffer, but we're still doing much better. So there's really something in the correlation structure in there, and we can push this further. Imagine now we have 50 randomly sampled voxels. So we really now really destroying the rank deficiency. It's not rank deficient at all anymore. Still, I think we get slightly, I mean, the PCA approaches the univariate performance. It's pretty bad, but we are always doing better, right? So for functional imaging data, this is usually, in my experience, has been the case, that PCA works better than univariate data analysis, at least for the kind of cognitive tasks that I looked at. For structural data, this has not always worked, and sometimes it hasn't actually. So if you take free surfer thickness ROIs or volumetrics, sometimes this doesn't work, just anecdotally. Okay, so I think I want to
question.
Yeah.
If you have data with absolutely zero noise, would PCA still perform better than univariate analysis?
That is a good. I don't. I would be tempted to say maybe not, no, but actually that's a good question.
Yeah. Sorry, I was asking the question because I was just kind of wondering if PCA is better than univariate analysis because you have noisy data. So the power of PCA is to distinguish the meaningful signal from the noise, or it's more than that?
No, I think that might be it. We could also later ask around. I think so, yeah. Because in other words, if there was no noise in the data, then yeah, the signature of the subtle distributed signal that helps me distinguishing where my naked eye or univariate diagnostic might fail, I don't think that advantage might slowly go away. Yeah. So I mean, it's a good question. I will simulate it. Yeah. So the broad takeaway points is that I think the SSM technique or PCA regression for functional data gets better held out data replication for prediction of outcomes, right? And this usually works in all the tasks, fMRI tasks I usually look at. This works. I can confidently say that multivariate usually works better than univariate. And then yeah, I mean, this just explains again what I showed. Then these caveats, I'd like to, this is maybe not so much an imaging feature, but I've seen it in other uses of PCA where people pick principal components in an isolated manner, and I wanted to warn against that. I mean, of course, you can do this descriptively, but it's maybe not a good thing to do from the point of view of inferential robustness. And I see this sometimes, with neuropsychological data or dietary data or sometimes genetics. So be careful with single principal components, particularly if they have lower variance contribution, right? Picking principal component 5 or 10 or 15, they become very variable even under resampling, and so they're inferentially not very robust. And then the last thing, I mean, that's just the point of pragmatics, because often you don't have, because we're dealing with many more voxels and observations, we're not there yet with population level neuroimaging neuroscience, although we're getting there with the UK Biobank. You often need to resort to non-parametric techniques like bootstrap or permutation tests. And now this is now trivial because computers are so powerful, but at an earlier time, people were suspicious about that. They wanted closed form solutions. They wanted to use parametric tests, but that's not possible. But I think it's not a big deal. And here you can see an example for one pattern that we derived, and here you see the bootstrap map, and here you see what you would get with FDR, and you see the two are quite similar actually in this case. Okay, so I want to come to the conclusion. I think we're running out of time. As I said, and this is now a lot more opinion, I mean, or sort of white paper kind of messages. I think multivariate techniques, and I'm particularly partial, as I said, to the scaled subprofile model. PCA regression marks the most basic complication of a traditional univariate analysis. And I think it's still worth doing, as it's a shallow technique saying it's one shot. There's no iterative fitting of any kind. And I think it's a very good reference standard also for deeper learning architecture. If you have convolutional neural networks, then I think PCA regression is a good benchmark standard that you should really try to beat. And it's also conceivable there ensemble techniques that are easy to do, like bootstrap aggregating, or boosting. So all of these early machine learning techniques would be easy to incorporate into this framework and might be a very good reference standard before you look to deep learning or convolutional neural networks or encoders. Oh yeah, and I said that here. Now to make it relevant to molecular connectivity, Ariana asked me, please bring it back to molecular connectivity, and I'm happy to do so. First, I would like to, ask people to look at this paper. This is Igor's paper, a review paper from 2018, and there's a really beautiful demonstration of variance concentration in the same people. So looking at the same people and I think this was an fMRI task, and this is resting PET, you can see that the variance concentration, when you look at the cumulative scree plot, is much better in the PET signal than the fMRI signal, right? We need 2 principal components to account for 50% of aggregate variance. And for the fMRI signal, we need 14 or so. So that tells you that multivariate analysis for PET makes even more sense than for fMRI, because the variance is so highly concentrated that it probably will work much better and give you much better inferentially robust results also for prediction out of sample. And then, as I said, I think when I think about how could this be used for the better ground truth of some of the traces of molecular connectivity, imagine we know there's some specific systems, you have the raphe nucleus or the nuclear accumbens, or locus ceruleus, for noradrenaline. Imagine you have this specific knowledge, then you could use the seed locations. That could be your endpoint signal for the SSM technique, and you can find sort of whole brain networks that are related. And in order to make it more related more to an endpoint like symptom scales or cognition, you would have easily tools like mediation analysis. You use Preacher-Hayes mediation. So you find, for instance, a brain-wide pattern related, let's say, to the raphe, for some serotonergic tracer, if it exists. I'm a little ignorant, but you'd find a whole brain pattern and then see whether that also can explain symptom scales or cognitive decrements. So that I think is a very simple extension. Okay, so thank you for your attention. I took a little longer than wanted, but hopefully this was clear enough. And any questions, if it can't be covered here, you can also email me and I try to answer to the best of my ability.
Thank you, Chris. This was a very good and very clear presentation. The session is now open for questions. If anyone has them, please unmute yourself and ask them, or you can also put them in the chat. So would anyone like to start with a question?
Yes, I have a
just unmute.
Chris.
Just unmute yourself.
Yeah. Hi, Chris. So I have a more general question about the interpretations of the PCA, the components, right? We have the components we can reconstruct it as a map, and for fMRI we usually interpret it as a connectivity, right? How do you think that it's related to connectivity, or in fMRI we also do a little further to have kind of a dual regression, some kind of how do you think that how interpretable, or whether we can interpret this as connectivity? How do you think about that?
Yeah. So let me go back to this talk. Sorry, now I'm not going to go into presentation mode, but I'm gonna. Can you see? Everybody can see this.
Yes.
So yeah, you're right. So what I have, and you're right. At some point, people were much more cavalier about the term functional connectivity, and even in the early 2000s, what we're dealing with here is, of course, just co-activation, strictly speaking, right? We didn't look at interregional temporal correlations, what's today called functional connectivity, for instance, or resting state connectivity, right? We did not look at connectomes, one per person here, right? Although these techniques work on connectomes also, but I didn't want to, overwhelm people, but they work arguably where you can treat voxels. You can treat edges in a connectome similar to voxels in co-activation patterns. But yeah, you're right. So strictly speaking, if we're going to be very legal about it, these are not connectivity in the sense of intra-subject connectivity. These are co-activation patterns, right? And I thresholded them here, right? I only, of course, there are loadings in every voxel in the brain. I only showed you the ones that are, significantly positive or negative. And what really happens when this pattern is expressed, activation in the positive loadings is that all the red areas show a positive correlation with one another. The green areas show a positive correlation with one another, but between red and green, it's a negative correlation. So the pattern activation in red and deactivation in green would happen in concert, right? So, but it's not connectivity. It's not connectivity in the sense of functional connectivity today. Then this question about dual regression, yeah, that's an ICA specific work. I guess we could take this pattern and do dual regression and get subject specific voxel-wise representations of this component. That always seems to me a little strange because we really want to, I'm interested in the group invariant pattern, but yes, I could look at how this pattern looks voxel-wise in each individual, but then we're back to doing subject-specific voxel-wise analysis, which is what I want to get around. So it's not so much done with this technique. So sorry, I'm talking along.
Thank you.
Yeah, sorry, you didn't, cover it, but like you say that then the component from PCA should not be interpreted as connectivity, but then what about ICA, since the component of ICA are exactly interpreted as connectivity, or resting state networks. So is it something justifying the different interpretation?
I know, but you have to now. I have to defend ICA a little bit. Very often, ICA takes the time course into consideration, right? Where, for instance, you could, I think, and I don't want to misspeak on such a big forum, so Vince can set me straight, or later. But often ICA is a multivariate decomposition that comes when subject and time points are concatenated together, right? So the question we had earlier when somebody asked me, I don't remember who it was, but you have subject variability, time point variability. I got rid of the time point variability by getting activation maps within subject, right? But ICA, it could be the gift package would work on where you pool everybody's time point over all people together, right? And do ICA decomposition on this whole data array. So now the time points, the time course information is in there also. So that's where maybe this interpretation of connectivity is a little more justified in this very orthodox sense, Yeah, any other questions?
So in the literature of fMRI, right now they also have this, they call it gradients. And basically it's just like the first PC, right? How do you think about that?
Yeah, the gradients is interesting, particularly these particularly, do you mean the Daniel Margulis work? Of these, yeah, that is a very derivative measure because I think you're using connectivity, right? And you are sort of making a statement, how does the connectivity change spatially, right? It's a real gradient in a spatial sense. So how it relates, I find it hard to think through how it relates to this kind of analysis here, right? I mean, you're saying that if you go from vertex to vertex, the connectivity profile to the rest of the brain, how much does it change, right? And if the gradient is strong, you get areas where this connectivity profile moves around a lot, So it's a somewhat more derivative analysis and uses real connectomes again, individual subject connectomes. So how it relates to this is a little hard to see.
Yeah, but I think what they have shown, for example, first of all, it is very reliable in a sense, or replicable. That, and also maybe theoretically they try to interpret this. The first, the dimension is that one is from a very like sensory motor, like
right.
a low level, and then there's higher
yeah.
level kind of the seems like have like functional maps. Maybe it's functional meaningful. That's what I feel.
Oh, I see. Yeah. Oh, I see this sensory periphery gradient to higher cognition that you get out of the gradient broke. Yes, it's interesting. Would it empirically look similar to some of these principal components? Yes, I think that's a very interesting question. And one, it's also testable, right? So in other words, does functional gradients coming out of connectomes, do they somehow look somewhat similar to task-specific components? Even over a variety of tasks. Yeah, that could be an interesting question. Yeah.
Any other questions from the audience?
Okay.
Oh,
everybody
doesn't seem like
sufficiently confused.
Or you were clear, then too clear, so there are no questions. Yeah, no, it's just that I see a lot of papers doing SSMPCA, and then frequently they, instead of creating a pattern in the end of combining components, they end up just using the first component because it's the one that explains most of the variants. That is something that I never agreed with. But now I understand a little bit more like why I don't agree, like my instinct was right. So, I mean, how do you discuss with this kind of people against using the first component alone instead of using a combination of them?
Actually, actually, I think the first component alone, I would have no problem. If you go back to, if we go back to the I mean, maybe there are better combinations that could be even powerful. But the first, if you go back, I don't know whether you see this, the first principal component actually wasn't too bad here, right? Also in my little, what really is problematic when you go lower down and pick single one, but the first one arguably also in my empirically here didn't work out too badly. So in this, I mean, the one thing is I would maybe, if they do SSMPCA and by what criterion they pick the first one, maybe they could do an even better job. But I wouldn't be too worried about the first one versus say number 5 here. Or imagine my correlation plot. Imagine you for some reason would have picked 11, right? Because it also looks pretty good. That would not be a good choice, from a point of view of robustness. There's also, I don't know whether Jim, are you on here? Do you want to say anything? No?
Easier.
Yeah.
Hi.
Regis. Hi. Good talk. That one thing that's important, that when I started using when I developed SSM, and it was for PET, and that what was so important there was that you had this global difference among subjects in the metabolic rate, and that people were arbitrarily using different methods to try to estimate that, and that there was no sense of how much variation at individual voxels or pixels there was that was accounted for by the global effect, and it was like 85% of the variance. And so if you made any kind of error in using other specific regions in order to estimate your global effect, thinking there was no specific experimental effect of interest, that you would be then getting an estimate of the global that would actually include the effect you were looking for. and so you're getting, you really suppressed the effects you were looking for in a big way, and that you needed a way of eliminating the global effect without having to make arbitrary assumptions, which is what SSM essentially did, assuming there was a global scaling effect. And so that's what makes SSM in a way different than PCA. And also the fact that you've really investigated in a very systematic way is what is the effect of rank deficiency, and that one of the original concerns about applying PCA to brain data where there were many more pixels or voxels than there were subjects in the analysis, that you know, why do you can no longer use the Wishart distribution in calculating statistics, and so people basically, you know, that itself really diminished what people thought was valuable about PCA. But then that was wrong, and that as computing became more powerful and you could use you know, the computer methods and unparametric methods to estimate significance and power and so on, then that, I think, got dispelled. And then just one more thing is that one thing that's often been a problem in neuroimaging is that they're one-shot experiments. You never try to replicate, and especially back then. And so what we started with PET was studying Parkinson's disease, where you had an animal model, so you knew whether or not you were on the right track by looking at the components that you were getting out, and and so that served as a very good way of deciding the value of the PCA method. And you know, you were able to make predictions of, and here we're talking about clinical data, so you were able to make predictions of severity of disease that were, you know, the correlations were 80 or 90% with the brain data that you extracted with SSM. So, and that was much larger than about anybody else was getting. So it's so, and it was I guess the last thing is that the concept of how the brain worked back then was that somehow there was a very specific pathway with a very few nodes in the pathway, and that I think we've come to understand that the brain process, you know, it brings to bear a lot of information from what you might consider disparate parts of, you know, of storage in the brain. And so really you needed the idea of multiple nodes as contributing to the, you know, behavior. And so, you know, it's those sort of things back then that were driving my interest in seeing what we could do with PCA. So, you know, that's just a short history.
All right. Cool. All right. Yeah. There's also a question about software support. I mean, I do have a package I can share. That the thing is, I almost think it's too much. It's in a way easier to play with MATLAB. Yeah, and I can give a few simple. I can even give a simple script, and then if you are reasonably familiar with MATLAB, you can do some of these things much easier rather than having to go to command line toolbox, and it's very clunky. And I actually don't use it myself very much. I do everything just with simple script.
I'm not sure how we are on time. Do we have time for one extra question?
I mean, I have time.
Yeah, I mean, we can ask, but if you have, if anyone has a question, we can ask.
Okay, so I have a question about the selection of components that you could use. So you had on your presentation, for example, the scree plot that you see that the explained variance by component, it goes down. How do you select the number of components that you would like to use in your model for prediction later? Because I've seen a little bit of everything. I've seen people putting thresholds on single component variance explained, or cumulative variance, or even just a random number of components. So what do you recommend?
Right. Yeah, that's a good question. That's a long-standing bugaboo, not bugaboo, but it's also always a point of discomfort. So I can only say, whatever you do, even if it's totally arbitrary, if you do it a priori and you don't snoop the data too much, I would have no problem with it. So if you say to yourself, I want to pick something that accounts for 50% of the aggregate variance, then that's fine. I would have no problem with that. So what I personally do use the archaic criterion or n-fold cross-validation. When the data are well behaved, the two give very similar answers in my hands usually. When the data are weird or funky, or there are unrobust points in it, or extremely influential data points, then I would rather stick with leave one out cross-validation. But I also have no answer. So this consumes whole careers, right? Subset selection, and there's a very good paper early on from Isabelle Guyon, a machine learning researcher from Berkeley, and she talks about the best selection schemes. But even there, that's a point of sort of discomfort. I don't think there's a priori a best way, right? So I mean, don't snoop around. I would say don't iteratively try to find things that work better. The archaic criterion, that's a one-shot deal, right? And it tries to take into account the bias variance trade-off. So it's a reasonable, principled manner. The Bayesian information criterion is extremely it imposes extreme sparsity. So I'm not too. It hasn't worked so well for me, but other people use that too, you know. So yeah, sorry, lots of rambling. There's no really satisfactory answer. You know.
Okay, great, thank you. Do you have any more questions from the audience?
There is one in the chat.
Oh, yeah, there's one in the chat. How do you feel this method? Oh, there's a method. Let me just open the paper.
Yeah, I don't, hang on, I need to pull it up, but yeah, you could ask me offline. Yeah, I haven't digested this method, but I'll be happy to read it.
Yeah, I guess this is better to have a separate discussion after reading the paper and just to have a discussion over it. But it's, I mean, always open to check new methods.
Yeah, I think in May we have a talk. I think it's about specifically for within subject, like within individual connectivity methods.
Okay.
We can discuss those kind of methods maybe later.
Okay. All right, guys.
Yeah, it will be the talk of Joanna Pereira in May, I think. So okay, so
okay.
Yeah,
you can ask her all the hard questions. Yeah, you could ask her all the hard questions to answer.
Okay, so then if there are no more questions, I guess we can close the session here.
All right.
Yeah, just remember to fill in our feedback questionnaire because we want to hear what you think about seminars, this seminar specifically, and if you have any suggestions of topics you'd like us to cover, we want to know. And so please fill it. Thanks a lot.
Thank you.
All right.
Thank you. Okay, Chris. Bye.
Bye.